So what I have for you today is a short talk on a cancer gene that you've probably never heard of. The gene or genes really in question are YBH1 in yeast and DUSP12 in humans. So these two genes, they are orthologs of each other, which means that they function basically the same. They just have different names in different organisms. And as everybody knows, people get sick. This, what we have pictured here is a liposarcoma, which is a cancer of the fatty cells in connective tissue. And just a little fact, in 2018, 18.1 million new cases of cancer were diagnosed and 9.5 million people died of cancer. This is just a little diagram from cells to DNA, the flow of information. And so we start off with this normal big cell. We go all the way down to DNA in the base pairs. And this is where mine and Dr. Beasler's research was focused. Specifically on this second step that you can see here, translation. So you have the mRNA, which is messenger RNA, and eventually, with the help of a ribosome, messenger RNA will create a protein, a final protein. And we were looking at, well, what if we mess with translation a little bit? So with a lot of genes, you can identify what the gene is and where it works, but not really how it works. And that's important for knowing what's going to happen to it if you mess with it. So here we have a normal, and then with one base pair change, we have a messed up protein. And we did this experiment in yeast, obviously because you can't experiment on humans. That's very unethical. So what we did, we used these yeasts that are very similar to humans. And what we were looking at is how do these key differences in proteins lead to a difference in normal cells and cancer cells. So this is our normal protein, right? And with just a little change in the shape, the entire cell gets messed up. And why is it so hard to kill these cancer cells? This is what we were trying to work around. So with chemotherapy, that targets all the cells in your body. That's why you get that nasty cancer sickness, your hair falling out and everything. And we were trying to figure out a way, well, can we target the cancer cells specifically so that we can stop those and not hurt the rest? And this is the protein in question, DUSP12. So this is the human ortholog, which basically it's evolutionarily conserved. All eukaryotes have it in some form or another. We did this through genomics and epigenomics and transcriptomics. So the combination of those lead to phenotype, which is the outward physical characteristics. And you need to know how something works so that you can use it for therapeutic intervention, which is treatment. Without knowing exactly how it works and where it works and everything, you can't use it to help people get better. And we did this by messing with the flow of information. So we would take a broken DUSP12 and insert it into yeast with a functioning YBH1. Specifically, we grew up some colonies from the mutants that we had, and then we looked at, okay, which of these are growing fine, like the wild type normal, which of these are growing a lot slower. And the slower ones are what we're really interested in. So the slower ones are what we sent off to be sequenced to see what actually makes them up. Here we have just a short video, if it'll work, of yeast growing. So you've got two, they both start off with just two cells and they're growing very quickly. They're growing at basically the same rate. So that's not what we would really think of as interesting. And this is the actual culture, one of the cultures that we grew up some preliminary cells in so that we could take a little swab of this nice big colony and move it into our controlled colonies. So why are we breaking the DUSP12? Well, if you have a car and you take out or break half the cylinders, it's going to run, right? But it's not going to run as well or as fast. And it's the same concept as YBH1. So you have this, we put a DUS12 into a yeast cell with YVH1, working YVH1, and when the DUS12 works, we didn't mess with it, the cell grows fine, nothing changes. But when we have a mutant DUS12, all of a sudden the cell grows a lot slower. And those interesting ones are the ones that we sequenced, like I said. And obviously this is kind of a crazy report. This is what we would get back after sequencing. Very difficult to read, even for me, and I had to read them quite a bit. But in the future, we hope to be able to take these broken DUS12s and insert them into cells with normal DUS12s and stop that cell from growing, specifically a cancer cell. So that's what we've got here. We have inserted green fluorescent protein into a normal cell, and that's what makes it glow. So questions. I'm going to have a couple minutes to answer maybe one, possibly two questions. But any questions that you might have, you can send to john.poe@lion.edu. I'm not a complete expert on the topic, so I may not exactly be able to answer your question, but I'll do my best. And then we have my bibliography. Just a few sources that I used. And I want to say a special thanks to Dr. Buser. Obviously, without him, I never could have done this research or this presentation. He's been a huge help all along the way. So yeah, thank you very much, Dr. Buser.